Okay, are you ready for quantitative reverse transcriptase rather, PCR? I know you are. So let's go. Let's talk about this and how it works. All right, so generally when you're doing reverse transcription, you're going to be doing it on messenger RNA. That's going to be the most common application. And what do we know about messenger RNA? What's it got at the front end or 5' end? Do you remember? It's called a cap structure. And a cap structure is it's an inverted G nucleotide that has an extra methyl group on it and an extra methyl group on the next nucleotide. That's what a cap looks like. But what does it have on the 3' end, the tail end? Do you remember that? And that's going to be a poly A tail, right? You remember that? Yeah, good. So we know that if we're talking about a messenger RNA, it's always going to have a poly A tail. And so even if we don't know what the sequence of that messenger RNA is, we can still make a primer that will bind it, guaranteed. And we can do that by just making a primer that is poly T, because T binds to A, and so it'll bind to the poly T or the poly A tail of any messenger RNA, and that's how we're going to get started. Reverse transcriptase is going to work like a polymerase. It can't start a copy on its own. It needs a primer in order to get started. But once you have the primer there, the reverse transcriptase can extend the primer. It's called reverse transcriptase because transcription is making an RNA copy from DNA, and reverse transcriptase makes a DNA copy of RNA. So it's kind of the reverse of transcription, reverse transcriptase. You get these reverse transcriptase enzymes in a number of viruses. Usually they're going to be retroviruses related to HIV. So there are going to be a couple little videos that I'm going to recommend watching, and one of those is going to take you through some reverse transcriptases. And the first one they're going to tell you about is MMTV, which is mouse memory tumor virus, which is an HIV relative. But there are many of these different reverse transcriptases, usually from viruses. And they can all essentially work the same. If you give them a template of RNA and a primer of DNA, they will faithfully make a DNA copy of whatever the RNA was. The copy is, just like when you're copying DNA, it's going to be a reverse complement. So if it says A in the top strand, it's going to say T in the reverse complement that you make. And so it's going to run backwards because two RNA or DNA strands always run in opposite directions. And it's going to have the opposite nucleotides there. But once you know the nucleotides, it's very easy to flip it back and forth. There are tools that will allow you to do this all over the place. Online, it's actually quite simple and a little bit of fun. Okay, so what do you need in order to do reverse transcription? Well, you need all the things that you would need for PCR, plus you need an RNA template, and you would need nucleotides, so DNTPs, deoxynucleotide triphosphates. You would need a buffer. You would need a little bit of water, just like before, and you're going to need a primer. Now, the primer for reverse transcription is going to be called the first strand primer, since we're going to make the first strand of DNA with it. And the first strand primer will often be just a poly T primer, because we know that's going to work with any old messenger RNA. And we'll just let the reverse transcriptase copy, starting from the tail end, and make a copy of whatever that RNA is. And then in the next round, we'll be able to go in with our specific forward and reverse primers and actually amplify this up. So, there's our reverse transcriptase bound to the end of the primer, and then it's going to copy and assemble all these little nucleotides to make a reverse complement of the strand that it is copying, just like so. And then once we have made a copy of it, once we have made a first strand, we can then heat up the mixture a little bit. As we're doing our denaturing step for PCR, our initial denaturing step, that step by itself will actually destroy the reverse transcriptase. And so we can just get it out of the picture so it doesn't complicate anything down the road. Then after that, we can just proceed to do PCR. And even though the reverse transcriptase is still in there, the enzyme is going to be unfolded, denatured. And so it's not going to do anything. It's just going to be laying there, as it's just some junk that happens to be in the same tube. Not going to affect anything further. And we can then begin to copy our complementary DNA and using regular PCR. And we will double the amount that we started with every single round. So 1 to 2 to 4 to 8 to 16 copies, just like that. Just like PCR and normal. And the way that we're going to quantitate reverse transcriptase PCR is by using a probe. So you could quantitate it using a cyanobromoine dye like Cyber Green. That does work. But the cyanobromoine dye will stick to anything. And so if you happen to have made a copy of the wrong thing early on in the process because you didn't understand exactly what you were putting, into the tube, if it was an unknown, for example, then you can show amplification, but it will be amplifying the wrong thing. And so if you want to make absolutely certain that the thing you are amplifying is exactly the thing you wanted to amplify, in addition to those 2 primers, you're going to have, rather 3 primers. First strand primer is up here. That's probably going to be your poly-T primer. That's going to be used for the reverse transcriptase. And then once that's done, we're probably not going to use that anymore. We just put a little bit of that in. And then you're going to have your 2 primers for PCR. You'll have your, I've drawn one here as black, the other one here is gray, as your forward and reverse primers that are going to copy the DNA. And you will have a third piece of DNA, fourth piece rather, 1, 2, 3. First strand primer, forward primer, reverse primer, forward primer. And the fourth piece of DNA is going to be a thing called a probe, which is, I hate to break it to you, just another primer really, but it's got 2 special groups on the end of it. One is a fluorophore, which is going to give off a fluorescent color when you hit it with laser light. So it will just shift the light, it'll shift the wavelength of the light to a different wavelength, and so you're going to put in blue light and it's going to show up as green light, something like that. Or you're going to put in green light and it's going to show up as red light, for example. On the other end of our probe, we're going to put a molecule called a quencher. And the quencher is going to be a molecule that can accept the fluorescence from the fluorophore, and it's just going to absorb it. It's going to absorb it, and it's going to give it off as heat or something else, and so you are not going to see fluorescence if the quencher is there. And the nice thing with the quencher is that it can actually accept fluorescence via resonance transfer. So do you remember resonance transfer? Just like when we were talking about photosynthesis? This is that little transfer where we're going to send electron excitement energy right from one atom to another atom. And we can do it right down a little chain, like this little DNA probe, from one end to the other. And as long as the quencher and fluorophore are attached to each other, the quencher will quench all the fluorescence from the fluorophore. So you can hit the fluorophore with your nice blue laser, and you're not going to see green light because your quencher is going to eat it all up, or eat most of it up. But as you do PCR, Taq polymerase has this neat property that when it runs into the back of a piece of DNA that's stuck to the template it's attempting to copy, it is going to chew off whatever is right at the beginning of this. It's going to try to basically pave the way for it to make more copies. And so by knocking this off, you're actually going to release the quencher. The quencher is going to go free. And now that the quencher and fluorophore are not attached to each other, you will now see fluorescence from the fluorophore because it cannot transfer by resonance transfer its energy to the quencher. The quencher is still floating around, but it can float anywhere, and chances are it's not going to be close enough to quench the fluorophore. So every time you make a copy, you're going to break one of these little probes, and so the number of broken probes is going to go up, and you're going to be able to tell how many probes you've broken because you'll have more fluorescence there. That's it. That's how a probe works, and that's how you do quantitative reverse transcriptase PCR. First strand primer, forward and reverse primer, and a probe, which lands right in between the forward and reverse primer. And as you copy it, boom, you release the quenchers and the fluorophore fluoresces. So, if you were to have a COVID test, for example, what you would get would be a reverse transcriptase PCR, a quantitative reverse transcriptase PCR. That's how the tests are usually done. And so they're going to be doing this on you, and I figure at some point you either will have already had this test done or will have this test done in the future. So it's worth understanding what this is. The way the test is run in the United States, they have what they call the N1 and N2 tests, which are just looking for different parts of the nucleoprotein gene, or the N gene of the virus. And so the nucleoprotein gene is this big long thing. I've shown you the protein sequence here, but remember protein is coded by RNA, and the RNA sequence would tell you to put in these amino acids. I'm just showing this because it fits onto a slide a little bit better, and you can actually read what what it looks like and kind of see how things are laid out. The N1 has a forward and reverse primer at the front of the N gene and has a probe that sits right in the middle. The N2 test has a forward and reverse primer back here and a probe that sits in the middle of those things. And there's going to be a first strand primer that's going to bind to the end of the N gene at the poly A tail, because the SARS coronavirus 2 has a poly A tail, just like a messenger RNA. That's just the way it happens to be built. So, it looks like I got my colors mixed up here. This should be a nice pink color, and we'll get to that. So if you're curious, these are the N1 forward and reverse primers. That's what they actually look like. You can see they're different lengths, but they're set up. This one has more C's and G's, and C's and G's bind tightly to each other. So these two actually have about the same melting temperature on them. And shown down here at the bottom, these are the N2 primers, forward and reverse. The N1 probe and the N2 probe will be the nucleotides that sit in between the N1 forward and forward and reverse primers for each set. And so as you're making copies with the forward primer, you're actually going to knock off the quencher from the end of the probe, and you're going to get fluorescence from the other end of the probe. Now, these areas are chosen because if we compare two different coronaviruses, so this is SARS coronavirus 2 on the bottom, and a different coronavirus you would find in a mouse. This is the mouse coronavirus, mouse hepatitis virus JHM strain, so that's why it's MHV JHM. That doesn't really matter, it's just showing you it's a different coronavirus. We've chosen areas where these viruses are going to be different so that you can specifically amplify just SARS coronavirus 2. We do this test because we want to know that you have a specific virus. We don't care whether you've had some other coronavirus in you, because that one doesn't matter. We care whether you've had the coronavirus that causes COVID-19 in you, if we're doing a COVID-19 test. And so we choose regions where the forward, reverse, and probe regions are going to be different, or different enough that we can bind specifically at each step. We would not want to pick a region like the one here in pink, because that's an area where if you put a forward there, a reverse there, and a probe in the middle, you would be detecting any coronavirus. And we don't want to detect any coronavirus, we want to detect a specific one. So that's how the people that are designing these tests actually work it out. And so to get a positive reaction, you have to have had the first strand primer bind, which will bind to any RNA, so any messenger RNA rather. So that's pretty easy. That's not too specific. You will have to have the forward and reverse primer bind, and you'll have to have the probe bind. And when you're making a copy from the end of the forward primer, you will have to run into that probe on the same strand. So you've got to have 1 first strand, 2, 3, 4 specific binding events happen, or else you will not get a positive. And normally they don't just run the N1 test, they'll run the N1 and N2 test on the same sample. And normally people would only give you a positive result if you are positive for the N1 test and the N2 test. So let's count up the points of specificity. You gotta have a first strand primer, which will actually work for both of these things. You've got to have 1, 2, 3, so 1, 2, 3, 4, 5, 6, 7 exact DNA matches to the complementary DNA from the virus in order to get a positive test. That's why we can be real sure that this test is very, very specific. So they test this out, they check it against other viruses, they check it against other RNA samples, and they have to prove before you can use it in the clinic that this is only going to amplify and give a fluorescent signal when you've got the virus in there that you're looking for. So there we go. That's how it works. And this is going to be as sensitive as PCR. So you can routinely pick up 50 copies or 100 copies or more of the virus. And that is very helpful when you're trying to track down a disease, for example. But like I said, there are lots of other ways you can use this. You can look at the metabolism of a cell, what genes are being expressed at a particular time. You can look at cancer. You can see which genes the cancer cell is expressing. That can tell you if this cancer cell is like a lung cell that is moved out of the lung and is, like lung cancer that is lodged in a different tissue. Helps tell you how you want to treat this thing. Quantitative reverse transcriptase PCR is just really, really useful and very widespread, and frankly, even fairly basic hospital pathology departments will have one of these machines in there so that they can do this technique, because it is, frankly, the gold standard for detecting not only what's going on, what kind of RNA was present, but checking how much of each RNA is present. So there you go. That's the story of quantitative reverse transcriptase PCR, and we built it up from first talking about PCR, then talking about quantitative PCR, and then adding on the reverse transcriptase before we did our quantitative PCR to make quantitative reverse transcriptase PCR. All right. So watch back through this if you need to, and there are those nice videos from YouTube there to help you out as well. Thank you very much.